Hello dear students, today we are going to discuss the topic aquaporins. The main objectives of today's topic are to study the brief historical background of aquaporins, to learn about how aquaporins were discovered, to learn about structure and function of aquaporins, to gain an insight into the role of aquaporins in osmoregulation, and to understand the various roles of aquaporins. Now, let's start with a brief introduction of aquaporins. Aquaporins are membrane water channels that play critical roles in controlling the water contents of cells. They are from a larger family of intrinsic protein that form pores in the membrane of biological cells, mainly facilitating transport of water between cells. These channels are widely distributed in all kingdoms of life, including bacteria, plants, and animals. Plant cells contain aquaporins through which water can flow more rapidly into and out of the cell than by diffusing through the phospholipid bilayer. In plants, aquaporins occur as multiple isoforms reflecting a high diversity of cellular localizations, transport selectivity, and regulation properties. Plant aquaporins are localized in the plasma membrane, endoplasmic reticulum, vacuoles, and plastids. In some species, aquaporins occur in membrane compartments interacting with symbiotic organisms. Plant aquaporins can transport various physiological substrates in addition to water. The important one for plants is the transport of dissolved gases such as carbon dioxide and ammonia, or metallides such as boron and silicon. Studies on structure and function of aquaporins are developed to address the molecular and cellular mechanisms of plant aquaporin gating and subcellular trafficking. Phosphorylation plays a central role in these two processes. These mechanisms allow aquaporin regulation in response to signaling intermediates such as cytosolic pH, calcium, and reactive oxygen species. Combined genetic and physiological approaches are now integrating this knowledge, showing that aquaporins play key roles in hydraulic regulation in roots and leaves during drought and in response to stimuli as diverse as flooding, nutrient availability, temperature, or light. A general hydraulic control of plant tissue expansion by aquaporins is emerging, and their role in key developmental processes like seed germination, emergence of lateral roots has been established. Plants with genetically altered aquaporin functions are now tested for their ability to improve plant tolerance to stresses. Now, let's move towards the history of aquaporins. The mechanism of facilitated water transport and the probable existence of water pores had attracted researchers since 1957. In most cells, water moves in and out by osmosis through the lipid component of cell membranes. Due to the relatively high water permeability of some epithelial cells, it was long suspected that some additional mechanisms for water transport across membranes must exist. In the late 1950s, Solomon and his co-workers performed pioneering work on water permeability across the cell membranes. In the mid-1960s, an alternative hypothesis, the partition diffusion model, sought to establish that the water molecules partitioned between the water phase and the lipid phase and then diffused through the membrane. Water molecules cross until the next interphase where they left the lipid phase and returned to an aqueous phase. Studies by Parisi, Edelman, Corvinus et al. Accentuated not only the importance of the presence of water channels but also the possibility to regulate their permeability properties. In 1990, Workman's experiments demonstrated functional expression of water channels, indicating that water channels are effectively proteins. Shortly after the discovery of aquaporins in animals, the identification of water channel proteins in plants was fairly unexpected. But it provided a strong momentum for studies on plant water transport. Early studies of these water channel proteins or pores called aquaporins contributed to a general boost in the molecular characterization of membrane transport systems in plants. After 20 years, plant aquaporins are recognized as multifunctional proteins transporting water as well as gases such as carbon dioxide, nutrients such as boron, silicon, or reactive oxygen species. Thus, aquaporin studies spread to many other fields of plant biology, from molecular membrane biophysics to cell biology and signaling. Their function also appears central for the physiology of plant growth and responses to abiotic stresses. Now, let's move ahead with a brief discovery of aquaporins. Until 1992, nothing was known about aquaporins. It was in 1999 when Peter, together with other researchers, reported the first high-resolution images of the three-dimensional structure of an aquaporin, namely aquaporin 1. Further studies using supercomputer simulations identified the pathway of water movement through the channel, which demonstrated how a pore can allow water to pass without the passage of small solutes. Agri was awarded Nobel Prize in Chemistry in 2003 for the pioneering research and subsequent discovery of water channels. Agri discovered aquaporins by serendipity. He studied the Rh blood group, antigens, and isolated the Rh molecule and a second molecule. This molecule was 28 kilodaltons in size and was called 28K. At first, they thought it was an Rh molecule fragment or a contaminant, but it turned out to be a new kind of molecule with unknown function. It was present in structures such as kidney tubules, red blood cells, and related to proteins of diverse origins such as in fruit fly brain, bacteria, the lens of the eye, and plant tissue. However, the first report of protein-mediated water transport through membrane was shown by George Benga in 1986, prior to Agri's first publication on the topic. This led to a controversy that Benga's work had not been recognized either by Agri or by the Nobel Prize Committee. Now, let's have a look over the structure of an aquaporin. The structure of aquaporins is highly conserved in plants, microbes, and animals. 4 subunits associate to form a single plant aquaporin, that is a tetramer. Each subunit has a molecular mass of 23 to 31 kilodaltons and spans the membrane with 6 alpha helices joined by 5 intervening loops. In contrast to the beta-pleated sheet structure of porins, these protein subunits fold within the membrane such that the hydrophobic amino acids are on the outer side of the pore and interact with the hydrophobic fatty acids of the lipid bilayer, whereas the hydrophilic amino acids are on the inner side of the pore and interact with water molecules as they move through the pore from one side of the membrane to the other. The amino and carboxyl halves resemble each other apparently, repeating a pattern of nucleotides. Some researchers believe that this was created by the doubling of a formerly half-sized gene. Between the helices are 5 regions, from A to E, that loop into or out of the cell membrane. 2 of them, B and E, are hydrophobic, with an asparagine, proline, alanine, NPA motif pattern. They create a distinctive hourglass shape, making the water channel narrow in the middle and wider at each end. Another and even narrower place in the channel, called the RR selectivity filter, is a cluster of amino acids enabling the aquaporin to selectively let through or block the passage of different molecules. Aquaporins can be open or closed to regulate the movement of water across the membrane. Gating is the term used to describe this regulated opening and closing of these protein channels. Gating through plasma membrane intrinsic proteins can be controlled by cytoplasmic pH, the concentration of divalent cations such as C2 positive, as well as by aquaporin protein phosphorylation. Another important thing in the structure of aquaporins is NPA motif. Aquaporin channels appear in simulations to allow only water to pass, as the molecules effectively queue up in single file. Guided by the aquaporin's local electric field, the oxygen in each water molecule faces forwards as it enters, turning around halfway along and leaving with the oxygen facing backwards. Why this rotation occurs is not entirely clear yet. Some researchers identified an electrostatic field generated by the two aquaporin half helices, HB and HE, as the reason. Others suggested that hydrogen bonds between asparagine amino acids in the two NPA regions and the oxygen in the water cause the rotation. It is still unclear whether the rotation of water molecules has any biological significance. Early studies speculated that the bipolar orientation of water molecules blocks the flow of protons via the Groth's mechanism. Groth's mechanism, also known as proton jumping, is the process by which an excess proton diffuses through the hydrogen bond network of water molecules or other hydrogen bonded liquids through the formation and concomitant cleavage of covalent bonds involving neighboring molecules. More recent studies question this interpretation and emphasize an electrostatic barrier as the reason for proton blockage. In the later view, the rotation of water molecules is only a side effect of the electrostatic barrier. At present, the origin of the electrostatic field is a matter of debate. While some studies mainly consider the electric field generated by the proteins half helices HB and HE, others emphasize dissolution effects as the proton enters the narrow aquaporin pore. Next important is RR selectivity filter. The aromatic arginine or RR selectivity filter is a cluster of amino acids that help bind to water molecules and exclude other molecules that may try to enter the pore. It is a mechanism by which the aquaporin is able to selectively bind water molecules hence allowing them through and prevent other molecules from entering. The RR filter is made of two amino acids groups from helices B, HB and E, HE, and two groups from loop E, LE1 and LE2, from the two sides of the NPA motif. Its usual position is 8 angstrom degree on the outer side of the NPA motif. It is typically the tight part of the channel. Its narrowness weakens the hydrogen bonds between water molecules, enabling the arginines, which carry a positive charge, to interact with the water molecules and to filter out undesirable protons. As discussed, lipid bilayers are quite permeable to water. Thus, there are two possible pathways for the movement of water across a membrane. One pathway through the lipid bilayer itself and the second pathway through an aquaporin. If this is so, why does a cell require any aquaporins at all? The results of recent experiments clearly indicate that the rate of water movement through a lipid bilayer with aquaporins is faster than a membrane that contains lipids only. Thus, the presence of aquaporins provides a low resistance pathway for the movement of water across a membrane. Furthermore, since aquaporins are gated, this provides greater control for the movement of water intracellularly as well as intercellularly. For example, the water permeability of the chloroplast is 2 orders of magnitude greater than that of the plasma membrane. Thus, this allows the vacuole to replenish or buffer the cytoplasm with water when the cell is exposed to hypertonic conditions. Thus, aquaporins are important in regulating the osmotic properties of plant cells. This process is called osmoregulation. Finally, let's talk about the functions of aquaporins. Addressing the cell autonomous functions, aquaporins provide a useful step towards integration of their role at the organ and whole plant levels. One major cellular function of aquaporins is osmoregulation. In agreement with the targeting of specific aquaporin isoforms to distinct subcellular compartments, water transport assays in purified membrane vesicles, isolated vacuoles, and protoplasts have revealed that membranes from a same cell can exhibit strikingly different permeability profiles. In tobacco cells, for instance, purified tonoplast vesicles were 100-fold more permeable to water than their plasma membrane counterparts. An extremely high water permeability in the tonoplast seems to be common to most of the plant cells. According to mathematical models, this property would provide the cell with the capacity of reducing peak fluctuations of cytosolic volume in case of a sudden change in extracellular water potential. Thus, plant cells use their large vacuoles as buffering compartments during cell osmoregulation. The delayed volume response of isolated protoplasts subjected to an osmotic challenge indicated that dynamic changes in water permeability may also contribute to plant cell responses to osmotic stresses. A direct dependence of water permeability on transmembrane osmotic gradients has been observed in tonoplast vesicles purified from wheat roots but not in tobacco suspension cells. The dynamic responses of aquaporins to osmotic stress have led to the idea that aquaporins localized on the vacuole or plasma membrane may serve as osmosensors in gourd cells during stomatal movements or in growing pollen tubes. Besides osmoregulation, plant aquaporins have many biological functions. Number 1. ROS detoxification and signaling. The role of aquaporins in physiological functions throughout plant growth and development are well known. As an integral regulator of plant-water relations, they are presumed to play an important role in plant defense responses against biotic and abiotic stresses. Consistent with the ability of tonoplast intrinsic proteins to transport H2O2 in yeast, transgenic overexpression or inactivation of these aquaporins can confer plant tolerance or sensitivity to oxidative stress, respectively. Among ROS, H2O2 has a relatively long lifetime, and its import by tonoplast intrinsic proteins into the vacuole may efficiently contribute to ROS detoxification. This implies, however, that a detoxification machinery is expressed in vacuoles. Up to now, most of these systems have been characterized in the cytoplasm. Plasma membrane intrinsic proteins can also have a significant H2O2 transport capacity, and the expression of some of these aquaporins is reduced by H2O2 itself. Considering that H2O2 produced in the epiplasm by combined NADPH oxidase and superoxide dismutase activities can serve as a signaling intermediate. Such signaling role, which has been established for mammalian, aquaporin 1, aquaporin 3 and aquaporin 8, still awaits experimental demonstration in plants. God cells which respond to abscisic acid through H2O2 production may represent an interesting model to address this issue. Number 2. Vehicular storage. The permeability of tonoplast aquaporins to compounds such as NH3 or urea has suggested a role for tonoplast intrinsic proteins in vehicular storage and remobilization of nitrogen metabolites. However, this role could not be validated in plants using reverse genetic approaches. In addition, plant membranes are equipped with other transport systems that mediate a more specific and vectorial transport of NH3, NH4 and urea. Thus, the role of TIPs in vehicular partitioning of nitrogen metabolites seems at most minor. Number 3. Chloroplast transport. Because of its roles in cell bioenergetics and photosynthesis, the plant chloroplast is a unique organelle in eukaryotic cells. One key reaction is oxidation of water to molecular oxygen that occurs in the thylakoid lumen. It was calculated that the consumption of water molecules represents 60 to 170-fold thylakoid lumen volume per day. Although NMR measurements show a moderate diffusional water permeability of the thylakoid membrane, a minute osmotic gradient might be sufficient to support the steady uptake of water required for thylakoid lumen refilling. Thus, aquaporins may be dispensable during this process. In contrast, the thylakoid lumen shows fast and marked volume increase in response to light, which may be facilitated by aquaporin activity. Carbon dioxide is another potentially relevant substrate of aquaporins in the chloroplast. Several physical and biochemical components of plant cells such as cell wall composition, carbonic anhydrase activities in the vicinity of membranes, or carbon dioxide permeability of the membranes themselves possibly restrict carbon dioxide diffusion throughout plant cells. The chloroplast envelope may represent a critical barrier limiting carboxylation reactions performed in the stroma through Calvin-Benson cycle. Aquaporins located on the chloroplast envelope may help dissipate ROS produced in the chloroplast under highlight conditions through H2O2 transport into the cytosol. This was all about today's lecture. Thank you.